seminar. My name is Scott, and I'll make sure that's working, and I'll be giving part of the presentation. Our first speaker is Nick Duesberry. He comes from the Van Andel Research Center in Grand Rapids, Michigan. So he's going to discuss his topic on controlling blood vessels. I'm going to let him go ahead and get started.
Thank you very much for the, can everyone hear me? Okay, thank you very much for the invitation to come back and talk to you. It's a pleasure to give you an update on the research that we've been doing for the last few years since we last talked. So the title of my talk is Controlling Blood Vessels in Disease, and talking about the insights that we're getting from the Italian Greyhound. Now, our focus isn't just on cancer, it's on a number of different types of vascular diseases. I'll tell you a bit about those in a few moments. So in this talk, I'm going to give you a very quick crash course in tumor vascular biology, talk a little bit about some of the research projects in the lab, but then delve in deeper into the insights we're getting into cancer and what dogs have taught us about cancer in people. And finally, I'm going to give you preliminary results from the Italian Greyhound Health Survey that we've been conducting with help and collaboration that have been part of the last 6 months. So, my lab for the last 15 years has been studying a single protein. That protein is called mitogen activated protein kinase, which is a mouthful, so we just call it MEK. Okay? And if you're a structural biologist, you'd be interested in its appearance. If you have a high-power microscope, this is what it would look like. But that's not really important for my talk. And this diagram here, I don't want you to memorize it. All I want you to see is that MEK is positioned right in the middle of a very large biochemical network that controls a lot of different activities inside the cell, inside cells. So it can help cells divide, it can help cells move around, and we think it plays a pivotal important role in blood vessel activity. And I'll show you the evidence for that in the next couple of slides. So one of the really important discoveries that my lab made about 10 years ago now was from a study where we injected anti-MEK drugs into mice that were growing a type of tumor called fibrosarcoma. We have these special mice in our lab. They're called athymic mice. And these mice have a very poor immune system. So you can actually take tumors from a human or from a dog, inject them into the mice, and the tumors will continue to grow. So that's what we did in this particular experiment. We took a human fibrosarcoma and implanted it into this mouse, allowed the tumors to grow, and then started treating them with anti-MEK drugs. And what we found was that while our untreated tumors continued to grow at a very quick rate, the tumors that we treated with the anti-MEK drug started to shrink very quickly. That wasn't so surprising because we knew that MEK was very important for controlling cell division. What was surprising was when we pulled the tumors out and had a closer look at them, we saw that those that had been treated with the MEK inhibitor had a very different appearance than those tumors that were not treated with the MEK inhibitor. So these, that maybe doesn't show up very well here, look very bloody and bruised. Those are the untreated tumors. But the tumors that have been treated with our anti-MEK drug look very pale, like they've lost a lot of blood. And that, in fact, was the case. When we, so this led to a hypothesis that we've been pursuing since then, that MEK signaling plays a crucial role in tumor blood vessel formation. We call it angiogenesis. This is a tumor that's growing on the backside of a mouse, and we're injecting a little agent to help visualize the blood vessels inside that tumor. I think you can see that you can see very clearly that there are blood vessels that spread throughout that tumor. When we take that same mouse and treat him with our anti-MEK drug, and then look at tumor blood flow, you see that the tumor now has a dramatically reduced blood flow inside it. And then if we look and actually measure the number of blood vessels that are inside that tumor, they're shown here in the brown color. So this is a microscope image of a tumor, brown with the blood vessels. These are our untreated mice. And these are the mice with tumors that have been treated with the MEK inhibitor. You can see that there's a dramatic reduction in blood vessels. So this led to a central concept then that we had in the lab that MEK signaling plays a crucial role in blood vessel function. And we're not only studying it in tumors, we're studying it in a number of different ways because blood vessels play important roles in a lot of different diseases. In tumors, in cancer, they're very important because they bring blood and nutrients into the tumor so that the tumor can grow. But it's also, blood vessels play an important role in a lot of retinal or eye diseases. So diabetic retinopathy, wet macular degeneration, retinopathy of prematurity, these are the leading causes of blindness in adults and in children, are all associated with abnormal blood vessel growth. Also, when you think about it the other way around, peripheral arterial disease that a lot of elderly diabetics get, where you get a decreased blood flow to your leg, is really the problem in reverse. So in that case, instead of having too many blood vessels, they have too few blood vessels. And finally, blood vessels also can serve as a site for the origination of tumors. So the type of tumor I'm going to be talking to you about today is Angiosarcoma, which of course you're all familiar with because it's a very important health problem in Italian greyhounds. Well, Angiosarcomas, or angiosarcomas as they're called in humans, actually arise from the cells that line blood vessels. So if we think that MEKs play a crucial role in blood vessel function, we think that by extension MEKs will also play a crucial role in tumors that form from those blood vessels. And I'll show you the evidence for that in just a little while. But first I'm going to tell you a bit about angiosarcoma in humans. So angiosarcoma is called a soft tissue tumor that arises from endothelial cells, and those are the cells that line the blood vessels. There's some evidence that angiosarcomas can develop as a result of prior treatment for radiation, let's say for breast cancer. Also after exposure to types of carcinogens like thorium dioxide or arsenic trioxide, which are found in insecticides, or vinyl chloride, which is used in some types of manufacturing processes. But nobody really knows what causes angiosarcoma. Nobody's identified any mutations, gene mutations that are associated with it, and nobody's identified the biochemical pathways that are critical for the formation of this tumor. However, this is a very important problem. So right now, the only way you can treat angiosarcoma really is by surgery, okay? And that's often followed by chemotherapy. However, at the time that most people are diagnosed, that tumor has already spread to other parts of the body. So while you can remove a lesion like this one, a tumor like this one, on an arm, generally within 2 years of that surgery, the disease recurs. And when it occurs at distant sites throughout the body, and it's usually fatal. Now, we have a problem. This is an exceedingly rare disease. So angiosarcomas themselves only account for about 1 or 2% of all soft tissue sarcomas. And soft tissue sarcomas only account for about 1% of all the tumors that people get. So this is a very rare type of tumor. There are perhaps only 60 or 70 cases in the United States every year. So for a researcher, this is a problem because I can't get enough tissue to work with. I can't get enough samples. From a patient's perspective, well, yes, it's a problem because you've got cancer, but also there aren't enough people doing research to try and come up with new ways to treat this disease. So these people are very underserved and are desperately in need of help. This is where dogs come in. So, one of the interesting things that I found when I first started getting involved in this was that dogs spontaneously develop certain types of tumors that also develop in humans at a much higher rate. So remember I told you that soft tissue sarcomas in humans make up only about 1% of all cancers. Well, the incidence of soft tissue sarcomas in general in dogs is more than 50 times higher. And that's looking across all humans. If you look at angiosarcoma, that's more than 100 times more common in dogs than it is in people. And again, we're talking about across all breeds. You know from your own experience that there are certain types of dogs, certain breeds of dogs, that have a much higher incidence of this disease than you get. So there's a wonderful opportunity here then to get samples that we can use for research to generate insight and develop new therapies for this disease that are not only going to help humans, but also help dogs. And that's really what my lab has been focusing on for the last little while. So in dogs, there are 3 different forms of angiosarcoma that form. Probably the most common type that we see in large breed dogs is a visceral angiosarcoma. So this develops in the spleen or in the kidneys, in the liver. And it's typically in larger breed dogs like golden retrievers, Clumber Spaniels, or German Shepherds. There's a cardiac form of the disease that appears to be higher incidence in Salukis. And then, of course, the cutaneous type that's commonly found in Whippets and Italian Greyhounds. And this type is particularly interesting for us because cutaneous angiosarcoma is the most common type of human angiosarcoma. These visceral types and cardiac types are very rare in people. This is the one that most people get. So, let's talk a little bit more about some of these diseases. Visceral angiosarcoma, as I told you, is the most common type in large breed dogs. It's a very sneaky disease. You often don't know your dog has angiosarcoma until the tumor has ruptured and they're bleeding out inside their abdomen. The dog looks very sickly, poorly. Take them into the veterinarian. The veterinarian will do an exam and tell you your dog has an angiosarcoma. And at that point, there's really not too much you can do because that tumor has already spread throughout the body. Now, this is an example of an angiosarcoma that we received in our laboratory. This is the spleen right here. You can see these tumors get exceedingly large. They're very fragile, so this is actually filled with fluid. And you can see how they easily rupture, and when they do rupture, there's a large amount of blood loss for the dog that they often don't survive. Cardiac angiosarcoma, again, is most common in dogs like Salukis, but it's also found in golden retrievers and in German Shepherds. And the early stages of the disease is actually an accumulation of fluid in the sac that surrounds the heart, and this can actually cause pressure on the heart that can lead to, well, obviously distress and anxiety, but can ultimately lead to heart failure. Again, an exceedingly poor prognosis and often metastasis at the time of diagnosis. And here you can see a couple of metastases that are present in the lungs from this patient. And then finally, there's cutaneous angiosarcoma, which is, as you all know, is common in Italian Greyhounds and Whippets. They can appear as single masses or as multiple small masses. This can be readily treated with surgery, and the majority of dogs that are diagnosed with this can actually survive many years afterwards. However, in some cases, when the lesion gets particularly aggressive, involving underlying muscle or starting to spread throughout the body, it's associated with shorter survival time. So, to study this, when I say we, I don't mean me, I mean Ro Frohman, who's a collaborating veterinarian and a consultant on this project, put together a canine cancer sample pipeline involving veterinary clinics across the United States. So there are more than 200 clinics that were involved in this project, each sending us samples back to Michigan. When we received these samples back in the lab, we put some parts frozen away for later study. We fixed some parts in a type of substance so that we can do pathology on the samples, and most of the pathology that we did is actually done by Dr. Angiosarcoma at Colorado State University. And then what we also do is we take some of those samples and we're able to put them into tissue culture flasks and grow them in the lab so that we can actually experiment with those tumor cells in the lab. I'll tell you a little bit more about that in the next few slides. So this slide is out of date, but at the time we put this together, we collected greater than 2,500 blood samples, predominantly angiosarcoma, lymphoma, and osteosarcoma, as well as melanoma. These samples were all processed to twice-lake blood. Here's a picture of Alyssa, who also joined us today, isolating DNA from some of these blood samples. And these samples are then shipped to the lab of Mark Neff, who's also at the Andel Institute. And Mark is working to try and identify genetic mutations that are associated with these diseases. What he's actually looking for are things called SNPs, or single nucleotide polymorphisms. And these are used to identify regions where there might be a mutation, and then we can go in and find that and to try and identify a single mutation in DNA that might turn a healthy dog into a dog with cancer. These are colorful examples of kind of results that he gets from the work that he's been doing. So this is called a Manhattan plot. It's called a Manhattan plot because all of these bars look like buildings in Manhattan. But each of these bars represents a dog chromosome. So chromosome 1, chromosome 2, chromosome 3. And then up on this side of the graph, we've mapped the probability or the chance that any particular chromosomal region is involved in disease. And the way this works is we just accumulate a large number of samples from dogs that have disease and dogs that don't have disease and look for differences between them. And as we accumulate differences in one particular area, that particular area can rise in its significance. So we have a threshold level below which we say, we really need more samples before we can tell you anything about this. And then once they get above this dotted line up here, that's when we really think they're worth pursuing and really focusing in on that particular area. So in this example of a study on Salukis, we have a couple of regions that look promising, but we really need to collect more samples before we can say we've identified a chromosomal region that's associated with cardiac angiosarcoma. Here's the work that Mark's been doing in the Italian Greyhound. You can see we have one that's definitely worth pursuing, but we've also got a signal that's way up on this chart that's very strongly associated with cutaneous angiosarcoma in Italian Greyhounds. Now, we can just tell you there's a region, it's on chromosome 3. It's a big region. It'll take us a while before we actually identify the exact location, but Mark's lab is working on that as we speak, so we're optimistic that if we can get more samples, we can narrow down this region and actually identify a mutation. In addition to collecting those blood samples, we're also collecting tumor samples, and this is the work that actually goes on inside my lab. So today we've collected more than 300 tumors. Well, this is at the time that this slide was put together. Our focus since this slide has been put together has largely been on angiosarcoma. So we've collected more than 100 angiosarcoma samples inside the lab now, and those are cutaneous, visceral, and cardiac angiosarcomas. And what we do with them is we actually use a special process to break these tumors apart into their individual cells, and we can grow them in culture. This is actually a photograph of angiosarcoma cells growing inside a dish in our lab. We can do a lot of different types of things with this. One example would be to try and treat them with different types of drugs to see if we can identify drugs that are at sensitivities of these cells so we can develop potential therapies. And that's one of the things we've been doing for the last little while. We've made some progress on it. So remember back to the beginning of the talk, I told you that my main interest for the last 10 or 15 years has been on this protein called MEK. So of course, because we knew MEK was important for blood vessel function, one of the hypotheses we wanted to test really early on was whether MEK was associated with angiosarcoma as well. Now, we did immunohistochemistry. So this is the same type of test they might do a tumor biopsy in a hospital, but we do it with the dog samples that we receive. One of the things we look for is something called ERK inside those tumors. ERK is actually a protein that's turned on by MEK, and ERK shows up as brown stain in these tumors. So you can see in visceral, cutaneous, and cardiac tumors, not all the cells show ERK, but a lot of the regions seem to contain ERK. So that's a good sign that MEK is probably active inside these tumors. When we take the cells out and put them into a dish, we can measure ERK in a different kind of way. This is called immunoblotting. And when we do that, we actually find that there's a large level of ERK in all of these different types of tumors. So that's also strong evidence that ERK plays a role, or MEK plays a role in angiosarcoma. what really helped to nail this down was when we treated angiosarcoma cells growing in a dish with a MEK inhibitor. So this is actually a drug that was used in phase 1 and phase 2 human clinical trials, and now that the drug companies don't want it anymore, they give it out for researchers, so researchers can use it. So we've been using it in our lab, and we've been able to show this is just a measure of cell health or cell viability growing in culture with increasing concentrations of drugs. And as we get a higher concentration of our MEK inhibitor, cells start to die off. So they don't like being treated by this MEK inhibitor. And one of the things we want to look for, of course, because we know MEK is important for normal blood vessels as well as for tumors, is a differential sensitivity. So we want a drug we can use in dogs or people that kills the tumor but doesn't kill normal blood vessels. So this is a cell line that's been isolated from a normal blood vessel. And when we compare the concentration of our MEK inhibitor that's required to kill the tumor cells to the concentration that kills the normal cells, we can see that there's a big difference. So while most of the angiosarcoma cells die when we treat with as little as 2 micromolar to 4 micromolar of this MEK inhibitor, those normal blood vessel cells require much larger amounts to kill them. So this is another indication that these tumors require MEK signaling. But most important are the work that we've done in the mice that I told you about. So we actually took some of those dog tumor samples that we received in the lab and implanted them inside those new mice, the athymic mice that had the compromised immune system, and those tumors started to grow. And the neat thing about this is, then this gives us an opportunity to test it in a real-life situation where a tumor is growing inside the mouse. These are the results of 2 experiments with either a cutaneous tumor or a cardiac tumor. And what's important to see here are the blue lines are those tumors in mice that have been treated with the MEK inhibitor, and the red and the green lines are tumors from mice that have not been treated with the MEK inhibitor. And I think you can see that the rate of growth of the tumors that have been treated with the MEK inhibitor is, for the most part, a lot slower than those that have been treated or were not treated with the tumors. And when we look at the size of the tumors, here we can see that they're substantially smaller than those that were not treated with either of our drugs. So it looks like in mice, when we grow these angiosarcoma, we can treat with a MEK inhibitor and we can actually reduce the growth rates of these tumors. And I think that's very promising. And then what was really exciting about this is we collaborated with physicians at University of Michigan and Memorial Sloan Kettering Cancer Center, and they worked with human angiosarcoma. We wanted to see whether this same pathway was active in human cancers as well. And here are some of the results from those kinds of tests. Now, because there are fewer samples available of humans, they can't do the same kinds of things that we're doing, but they did similar things. So remember, I looked at P-ERK or ERK inside the tumors. They did the same thing and found that half of the tumors that they tested actually had P-ERK inside them, just like the dog tumors. They looked at gene expression inside those human tumors, and they found that there were a number of proteins, shown here in these red dots, that were active in angiosarcoma. These red dots correspond to proteins that are turned on by MEKs. So a number of proteins that are turned on by MEKs that are present in the angiosarcomas, but not in adjacent tissues, so blood samples, kidney samples, or muscle samples. So this suggests that MEKs are actually active inside these tumors. And this is just a confirmation test that shows that when we look at one of these factors, MEK, you can see by that pathology stain, we can see them both in human samples and in dog samples. So this shows that there's that the canine samples were actually able to give us some really interesting insights, not only into the biology of angiosarcoma in dogs, but also into the biology of tumors in humans. So just to summarize the first part of what I've told you, the incidence of angiosarcoma is increased in purebred dogs. We found active hurt in a large majority of canine tumors that we've examined. And those tumors seem to respond to MEK inhibiting drugs, both in vitro and inside mice. When you look at the tumors, the pathologist looks at the tumors, they can distinguish between canine tumors and human tumors. And at a biochemical level, canine and human tumors both have active hurt present. So there's a high degree of homology or similarity between these tumors. But there are a lot of questions that we're still trying to answer. Not all of the cells have active MEK or hERG inside them, so which are the ones that have the active signaling in them, which don't? Are MEK good targets for treatment of angiosarcoma in humans and in dogs? And can we design new approaches, combinations where we use a MEK inhibitor plus a different type of inhibitor to get a more effective strategy for killing these tumors inside humans and dogs? And what are the best approaches for our follow-up studies? So we actually have several of these ongoing right now. Nick Anderson and Alyssa, both in my lab, are working on new strategies for combined treatment with different types of drugs to block the growth of Angiosarcoma. We put together a collaboration with scientists at our institute, as well as veterinarians at Michigan State University, and leading oncologists at Memorial Sloan Kettering Cancer Center to design a grant application that's been submitted to the AKC Canine Health Foundation. We should hear back from that, I hope, within the next 2 or 3 weeks. But this is a project to examine canine hemangiosarcoma at the Andel Institute, translate the results from those studies to human cancer samples at the Memorial Sloan Kettering Cancer Center to see whether we can come up with better therapeutics both for dogs and for people. And then finally, the last project I'm going to tell you about that's ongoing is the Italian Greyhound Health Survey. And this was spearheaded by Bo Froman, who's here in the audience, as well as Sheila Wozlowski, who's a University of Michigan undergraduate student who worked in my lab this summer and put together a lot of new information with the slides that I'm going to show you in the next little while. So you're all familiar with this survey. It opened in January of this year and closed in May, although we still did have a few entrants coming in after that, which is fine. We were really surprised at the number of people who participated in this. I have to admit, going into it, I wasn't very optimistic. I thought at best we might get 100, 150 people. But 700 people responded. I mean, this just, it was phenomenal. We were so excited every day because we come into the lab and then go up again. And this is what we need. We need these large numbers to make any kind of inference from the information that we receive. So, you know, the survey, we looked at a number of different key health problems or concerns of the for Italian Greyhounds, dental records, information about cancer, fractures, spinning habits, sunbathing, seizures, and a couple of other different things. Today I'm going to focus on some preliminary results regarding the cancer studies, sunbathing, and seizures, and I hope to be able to give you a more complete report as soon as we can, just to understand that we don't have enough people in the lab to do this, but we're working as hard as we can to try and move it forward. So, cancer frequency. Of the 700 dogs that we looked at, at least 113 of these had at least one of the 6 following cancers: lymphoma, mammary cancer, melanoma, mast cell tumors, angiosarcoma, and angiogenesis, which is a benign disease or benign lesion. So approximately 15% of respondents indicated that their dog either had or has cancer. When you break it down by tumor type, it turns out that hemangiosarcoma make up almost 1/3 of tumors that are reported for Italian Greyhounds, followed closely thereafter by hemangiomas and mast cell tumors, mammary cancer. This is a really interesting spectrum of disease and contrasts sharply with what we see in golden retrievers. So in the last published study, we can find in golden retrievers, which was done a fair while ago, Angiosarcoma actually forms a much smaller proportion of disease, so it makes up only 16% of cancer that golden retrievers get. And then what's even more interesting is that while golden retrievers predominantly have a visceral type of disease, it's clear from our Italian Greyhound health survey that 90% of the tumors that were reported for Italian Greyhounds were the cutaneous form that's most common. There are a lot of anecdotal reports. I couldn't find a good one for Italian Greyhounds, I'm sorry, but if you've got a nice picture of a sunbathing Italian Greyhound and you don't mind it used, please send it to me. I'd be happy to use it. So there are anecdotal reports that are out there. I'm sure you've heard them. That there's a link between sun exposure and Angiosarcoma. And you know Italian Greyhounds love to sunbathe. So one of the questions we had was, is sunbathing associated with incidence of this disease? Is it possible that ultraviolet light may be one of the triggers for an angiosarcoma? So we compared the sunbathing habits of dogs without cancer to dogs with either angiosarcoma, angiogenesis, or some of the other cancers. And what we found was that there's actually no difference between any of the groups. So approximately 70% of the dogs in each group love sunbathing. So that's no big secret. Italian Greyhounds like to sunbathe. So we thought, well, maybe it's not just that they like to sunbathe. Maybe it has to do with the amount of ultraviolet light that they're exposed to. So we broke things down by regions. We had this map that we can map or show the aggregated UV exposure in different regions of the country. We took the survey results and mapped them onto that. What we found was that, with the exception of angiomas, there's no apparent link between UV exposure and the incidence of angiosarcoma, mast cell tumors, melanoma, mammary cancer, or lymphoma. Now, for angiomas, what we saw was that in the north, they had 30% less angiomas than we would have expected based on the exposure. And in the south, they had 30% more than we would have expected just based on chance again. So there may be a link between UV exposure and angiomas. However, our survey didn't take into account movement of dogs from one region to another. And we don't really have accurate measurements of UV exposure in these areas. So I think before we draw any firm conclusions, we want to do a follow-up study and examine individual exposure to UV light in dogs and see whether that's linked to angiomas at all. Now, this was perhaps one of the more curious things that came out of this. There appears to be a link in Italian Greyhounds between cancer and seizures. Let me explain that. So, your own website lists idiopathic epilepsy as a health concern for Italian Greyhounds. So, we asked owners about this. In our survey, owners reported that 97 dogs, or approximately 14% of all the dogs that were included in the survey, had experienced one or more seizures. Now, Rowe noticed, and it's really interesting, that 29 of those dogs, or approximately 30% of the dogs that had seizures, also reported having cancer. That compares to the 15% incidence of cancer in the general population. And this seizure canceling appears to be strongest for angiosarcoma and mast cell tumors. Dogs having one or more seizures had more than twice the incidence of these tumors compared to the rest of the population of dogs. Now, we have absolutely no idea how seizures are linked to cancer. In human populations, of course, seizures are associated with cancer, but typically with brain cancers, not with cutaneous cancer like angiosarcoma. There's a possibility that there could be some kind of, that there could be a non-causal link. So you might imagine that in an older population of dogs, there's a greater chance of cancer and a greater chance of seizures, but idiopathic epilepsy has an earlier onset than you'd expect to have that kind of association. So again, follow-up studies are warranted to see if there is a positive link between these two conditions. So where do we go from here? Well, there's several questions that we haven't answered that we'd like to answer. Why did Italian Greyhounds develop cutaneous disease while other breeds develop visceral or cardiac disease. Is this one disease, or are we looking at a couple of distinct diseases? We really don't know. I think this is important to find out. What are the genetic causes of Angiosarcoma in Italian Greyhounds? And can we find the similar lesions or mutations in humans? Can we develop tests that can predict who's at risk of developing skin lesions? If we can identify a gene that's associated with angiosarcoma, then you can start testing at an early stage so that you'll know whether your dog has a predisposition to develop these lesions, and you can be a bit more vigilant, looking more carefully to see whether they show up or not. Can we identify factors that cause those early lesions to become more aggressive and to spread throughout the body? And of course, how can we treat this disease? We think that MEK inhibitors are part of the answer, but as I've indicated, I think we can actually come up with something better. We just have to look for the right combination that works in partner with that MEK inhibitor. And I think your involvement, the involvement of the Italian Greyhound Club of America is going to be critical to this. I always tell owners of the clubs that we need your blood, sweat, and your tears. We need your dog's blood because we need to identify or get more samples so that we can identify these mutations that are associated with the disease. Sweat is really just getting the word out there, letting people know that there are researchers who really would like that material. There are ways, and once the samples have been removed, they don't have to get thrown in the garbage. Send them back to us so we can actually do something helpful with them. And then finally, tears. If, heaven forbid, your dog does get sick and ends up having to be put down. But if that does happen, please consider sending the tumors from your pet to us. They're really helpful for our research. And finally, I just want to acknowledge the people who've done this work. Most of the projects I've told you about are actually done by this group here. It's an effort led by a postdoctoral fellow, Dr. Nick Anderson, who works in my lab. Also, Roe Bowman, veterinarian. Sheila Wazowski, an undergraduate student from the University of Michigan. Alyssa Wazowski has done a lot of the tumor sample preparation and all of the work that we do in our mice. And then we've got a lot of wonderful collaborators around the country. Larry Baker, University of Michigan, Gary Schwartz, who's at Memorial Sloan Kettering, Barb Kitchell at Michigan State, Ben Kampstock, Colorado State, Brian Nicklow, Kyle Birch are both investigators who are at the Institute and help us out as well. So I'm going to stop my talk right there, but I'll take any questions that you have. Go ahead.
A couple questions. When MEK inhibitor was stopped, did you see any recurrence of angiogenesis?
So in the tumor models, we haven't actually followed them out, but that's one of the things that we want to do. So because samples are limited, we did all our initial analysis on the tumors at the end of treatment. But in the next round of experiments, one of the things we will be doing is stopping treatment to see whether the tumors grow back or whether they stay low. We have done those kinds of studies in melanoma, which is another type of tumor that appears to be very sensitive to the MEK inhibitors. And in those cases, once we stopped tumor treatment, we followed them up for an additional 30 days and saw there was no regrowth of the tumor in that period. We haven't followed it out later.
So my next question would be status and impact on longevity beyond the treatment. You don't know.
On
Longevity.
For dogs, we certainly don't have
But for
it.
the mice, but we don't know when treatment is
Well, again, most of the mice, we can't just keep mice around all the time. Once the experiment's over, we have to move them out so we can move more mice in. I'm optimistic that it's going to prolong survival in dogs that have been diagnosed with angiosarcoma. And that's really one of the things that we hope to answer with that proposal that we have in with the AKC right now. So if any of you have any friends in the Canine Health Foundation, call them up and say, these guys really want to get this done. So we'll be doing that work at Michigan State University if this goes through, initiating clinical trials in dogs to see if we can actually prolong survival after a section.
Any viral involvement?
Absolutely. So this tumor very closely resembles one other type of human tumor called Angiosarcoma. Angiosarcoma is actually caused by human herpes virus. Okay. So there's a distinct possibility that Angiosarcoma could also have a viral component to it. However, no one's identified it. I don't know if there have been any intense investigations of it. Again, the problem is that there are so few samples that are available. But there are tools that can allow you to do that now, I think a bit more easily than it could have been done 5 or 10 years ago. And that's certainly something we would like to do. Again, not required. That's that.
Do those tumors ever develop a resistance to your MET inhibitors?
It is entirely possible that they would. Tumors are really they're tricky things. So if you look at a textbook, they'll show you a picture of a tumor and they'll show it's very homogeneous. It looks the same all around, but in reality, it's very different. What it actually is a series of different types of mutants, all contained, or mutant cells contained within one tumor. So you can develop a treatment that will knock out 90% of those tumor cells, but the 10% that it doesn't knock out regrow, and they're resistant to that particular type of drug. So this is actually one of the reasons why we're coming up with this dual approach to treating the tumor. So we don't want to treat with just one drug. If we treat with 2 drugs, we think we've got a better chance of blocking any kind of resistance from forming, or actually pushing it out for a longer time. Maybe we have to use 3 drugs. I don't know. So a lot of the stuff on melanoma right now that's working with MEK inhibitors is actually finding exactly what you indicated. So they treat with one drug, they show a miraculous result. All the tumors melt away, but then about 6 months later, all those tumors just boom, come right back all at the same time. So they develop resistance, they don't respond to that initial drug. So now they have to go in with or they're trying to identify a second line drug that they can go in with and try and knock down the tumors again. Thank you. Yes?
One of the items that was on survey was on the consequences,
Okay.
which is also a vascular disease that causes that usually really died. For some unknown reason, the blood supply gets cut off, the blood dies, and one of the consequences of the mom.
That sounds very interesting. I'm not familiar with it myself, but I'm sure Ro's going to tell me all about it.
It's not we don't see it a lot in attorney pregnancies, but it does occur. I went through a little literature on it,
Okay.
but I was just wondering if this could eventually in the future lead to
Well, I mean, that's what we're hoping to do in that peripheral arterial disease study, really, is to find ways of reversing or restoring the body's ability to reform blood vessels when blood vessels get occluded. And it turns out that patients that have advanced diabetes, they have a very limited capacity to regrow blood vessels after a vessel becomes blocked. So what we're investigating is things that are downstream of MEK activation. And if we can find out what MEK turns on, then maybe there's another way that we can turn it back on so that we can restore blood vessel formation.
Yeah.
So perhaps that's in parallel to what you're
and
talking
this
about.
starts to happen in very young dogs.
That's interesting. I mean, one of the things that I'd like to talk with you about at some point is a follow-up to the health survey that we've done. I don't want to start it before we finish this one. But I think when we get to the end of it, we sit down and start talking about the results that we got. We'll certainly have questions that we wish we would have answered. I think maybe the Italian Greyhound Club will have had questions that they wanted to have answered in that. So maybe that's when we need to start thinking about a health survey too. And maybe 6 months, a year from now, we can throw a survey back out there again. Maybe we get more respondents and answer some of those questions that we didn't get the first time around. Any other questions?
I have one.
Yeah.
Do we have any preliminary percentages on the autoimmune diseases?
Ro, did you?
That was, unfortunately, we did this with a Google poll, and we tested it, and then we released it live. We didn't realize until we were about 600 dogs into the responses that one section didn't work like it should have. And that was the multiple choice section where you could say, does your dog have this, or this? And it was supposed to be able to accept yes as an answer for every single category, and it did not. And we didn't know that until we started really crunching through the numbers. So that's definitely something, and it was a lot of the things that you guys were really interested in, so we felt terrible about that. There was no way to go back at that point and re-put that out there, but it's something that we really hope to be able to incorporate into a further follow-up study. So our apologies, something we all learned the hard way. So the numbers we did get were for about 100 dogs, and they're not reflective. So we didn't want to put them in at all because we know that they're not accurate at this point. So, but we will do this. So.
Any other questions? Okay, thank you very much.